Now we are recording. For anybody watching the video at a later date, I'll remind you, this is YouTube, so you can speed up the video. At the bottom right of the screen, there should be a little gear icon. If you click that, you can change the speed. You can speed it up, slow it down, whatever you need to do. Since there are already 3 people online, and usually online people don't get as much extra credit, I'll go ahead and do the first word for attendance. And this is an extra credit word. And it is carpet. Like, I want to get new carpet, but I'm going to wait until my kids are older because they are currently destroying my carpet. And obviously, we still have about 15 minutes. So anybody who's watching this as a video instead of a live stream, you might want to fast forward until you see me project something on the screen. Morning. So, like I said, Durgan is here, or online. Cook is physically here. Burton is online. Hughes is online. And Hannah is online. So I do plan to hold online office hours today, but finally, I've had this thing since 2016. Anyway, I'm scheduled to get a new laptop from the university today. So if they're doing the switch over during office hours, I'm obviously not going to be online for office hours because I won't have a computer, just so you guys know. Excuse me. Yeah. Yes. Well, it works sometimes, it works doesn't. Or sometimes it doesn't. My recommendation has been to get it done beforehand so you don't have to come in. But if you can't, just come in and do the best you can. That's really the only option, other than, like I said, doing it somewhere else. But if you do it during lab hours and you need help, obviously you won't be. If you're doing it somewhere else, you won't physically be there. But you can still contact me or Elijah if you need help, and we can meet you online. Yeah. Unfortunately, I can't think of any better option. Other than do it before you come in, or fight with the internet. I wish I had something I could do better about the internet, but it's completely out of my hands. And moving forward, I think all the labs are online because it's just not practical to do a real experiment anymore in person. Like we can't simulate, or we can simulate. What we can't do is actually like an experiment for natural selection, for example, right? We can't see evolution happen in real time. So we have to do a virtual lab. But yeah, let me keep brainstorming and see, maybe I can talk to IT people, see what's going on with that. Anyway, this week's lab is very easy. Speaking of which, the first part you have to do here, well, if you haven't done it at home, and that's just doing like a simulation of DNA extraction. And I actually find it better than the real hands-on one. The hands-on one's good because you get to do it hands-on. But I've noticed historically when they do the hands-on ones, just like a lot of the other labs, they're so focused on the details that they miss the big picture. But when you do the virtual version, it forces you to look at the big pictures. Like, first we mix this with this, and the reason we're mixing this with this is because this thing does that, right? And then you have to answer questions about it. So that's the plan. I'm sorry for the inconvenience. Do you have any ideas? Yeah. If you come up with something, please let me know. Yeah, that's also a good idea. Well, that's actually why, like on Monday, I know it sounds silly, but Monday I went to Roca's and invited people to come to that. Said, bring your stuff to me, I'll be there. That way we can have a little bit of fun and do it somewhere else. But yeah, I'll do that more often now that I think about it. Maybe I'll tentatively try every Wednesday to go somewhere for lunch and then just invite people who want to come if they need help for labs. Try to change up the location every week. It's a good idea. Also, there's 2 different Wi-Fis in here. Maybe we'll make sure you get the right one. I need to look into that. Because, once you've logged into it, you're automatically logged into it. So you forget how to log into it because you don't have to do it every time. What, like right now, are you connected to a Wi-Fi?
Here,
yeah.
It shows the networks, right? But
yes.
If you try to get on them, it does not let you. And then occasionally they all just disappear.
Interesting.
I can show you. I think they're online right here.
See, I'm on WVSU Wi-Fi, and I find that one better than WVSU Wireless or WVSU Wireless Fast.
So it's like this.
Uh-huh.
So I click on that. Well, now.
That's how it goes, right? What you try to show somebody. Yeah, I know how it is.
Oh. Okay. It was just flickering there.
Yeah. Try WVSU Wireless.
It makes, every once in a while, it makes a copy of it for somehow.
Interesting.
It connects.
Interesting. That's also interesting. You should ask you for credentials to get on that one.
And
It's
then it
annoying.
disconnects again.
Good grief. And mine's disconnected.
And every once in a while, again, they all just disappear. And sometimes they disappear for 5-10 minutes.
Good grief. Yeah, I don't know what's up with that.
And like I said, every once in a while, when you click on them, it shows up as it's connecting. But the network is still there in the My Networks too. But the one that you're connecting to has 1 bar.
Oh,
the one that
right.
it's connecting to has 3. Just makes a copy, and then they all disappear.
That is weird. And I've never seen that with my phone. But like I said, I don't log on. I don't look at the log on very often because I'm just automatically long dog. Yeah, that one does require a password though. The one WVSU Wi-Fi requires a password. So that's the one.
I've never got that.
It's let me think.
I can ask somebody. I have one of my relatives is Belinda Fuller.
Yeah.
She's mine.
Okay.
I can ask her about it. I just haven't.
Yeah, it's easy. I just can't remember it. It's something like yes. That's right. Yeah, you do that, and then it'll prompt you to put in your like your email, and then your password. Yes. So go state, and then followed by that. Yeah. Thank you. Yeah. Hopefully. All right. Let's see here. Oh, we've got more people. Let's do another extra credit word. Actually, for you in person, this won't be extra credit again today. We had like a couple people who, out of the 6 words, they got 2 of the words. So let's just try to reward those who are paying attention as they should. So anyway, this one will be extra credit, and it will be let's see, sticker. Sticker, like when your kid goes to the dentist, they give them a sticker sometimes. Same thing as doing the same thing? That is so weird. I don't know. Because mine's never done that. Like, I know when I've been signed out of that WVSU wireless and I've had to use the other ones, that one's giving me problems. But off the top of my head, I don't think WVSU Wi-Fi has. I could be wrong. And then when I'm in my office, I'm physically plugged in, so I'm not using Wi-Fi. What a pain. I wish I could help. So let's see. Fisher is online. Mullins is online. Kincaid is online. Tate is online. Lewis is online. Crowder is online. I think I have everybody online, at least for now. All right, less than a minute. So we'll go ahead and do official attendance. So again, if you're online, if I say you're not here and you're online, don't worry about it. Just send me the words after 9 a.m. For the rest of you, I know. Well, I'll wait till everybody else gets here to say that. Anyway, let's do some attendance here. Abigail is here in person. Kincaid is online. Andrea is not here. Mullins is online. Lewis is online. Cooper? Did I say Cooper? I did say Cooper. Thought I said Cooper was online, but maybe not. Anyway, Hunter is here. Yeah, Hunter is here. In WVSU gear. Yeah, make sure if you're in WVSU gear, let me know. Get you a little extra credit there. Caleb's not here. Crowder's online. Reinhardt. No. Isabel's here in person. Julia. Kennedy is here in person. Hannah's online. Larissa's not here yet. Madeline is here. Hughes is online. Galloway's not here. Cook is here in person. Durgan is online. Tate is online. Burton's online. Kinder? Not here. Jones? Not here. Elias is here in person. Zach is not here. Fisher is online. It is 8:01. We'll get started. So for those of you in person, today will be another day where the words are not extra credit. It's just for attendance. So make sure you write them down, whatever you need to do. Anyway, and the first official word, let's say date. Man, I hate that screen. Anyway, really quickly, with some announcements. So lab report, I got a great question about the lab report. And I've reminded a few people in writing, but not in person. I keep meaning to do that. So the lab report, again, I'm trying to hold off on gradings because I want your TA to get some experience. Plus, the longer, especially when it comes to this lab, the photosynthesis/respiration lab, the longer it takes for you to get it graded, the longer it takes before it's due. But you don't have to wait for it to be given back to you to do the lab report. You can do the lab report now. You could have done most of it before we even did the lab, right? The introduction, the abstract, and then maybe you might have to make some changes after you actually did the lab. But go ahead and do the lab report. You don't need to wait for it to get back. It's just that's when it's going to be graded, right? Or excuse me, it's going to be due 2 weeks after it gets back. So yeah, go ahead and do the lab report. Let us know if you need help. Again, I'm trying to do a little bit less grading in the lab. So yeah, any questions about the lab report? All right, and also I will tell you, like I mentioned a long time ago when we first started talking about the lab report, I said this and I'll stick by it and remind you and myself, I'm going to be less picky with the due dates on the lab reports. Independent work's coming up, which I'm about to talk about here in a second. So yeah, basically what I'm saying is if you find yourself like, oh my God, I'm in a crunch, I need to do my lab report, I need to do independent work, whatever it is, I would say put the lab report at the bottom of your priority list. You good? Okay. So that's it for lab report if there's no questions. This week's lab, I'll remind you, if you've already done it, you don't need to come in. Normally, I know I've been saying, I'll tell you specifically, one-on-one that you don't have to come in, but this week I'm saying don't worry about it. If you say you've done it, you don't need to come in. Just make sure you do both parts. Make sure you do the simulation at first and then the video after a second. Any questions about this week's lab? All right. One of your classmates made a good point. Like the Wi-Fi in this building does stink. And I wish there was something I could do about that. But unfortunately, there's not. Anyway, any questions about this lab? All right, exams. First of all, now that we're done with exam 2, that was quite a mess. But we're done with the exam 2 officially. So that means I can now meet you online and give you exam 1 feedback, which would help so you know all the correct answers so you can study for the final exam using exam 1. And also, as this is the case with any assignment in this course, if you meet me online for feedback, you get at least a 5% boost to that grade. So any questions about exam 1? Exam 2, what a cluster this has been. I don't know what's going on. This is like IT stuff that I'm trying to figure out, or I'm trying to get help with and figure out. It's closed. I can see where your right answers and your wrong answers, but it's not giving me the grade. Now, I know I can go in there and do it manually, and I'll do that if I have to. But I know there's also some sort of efficient way to do it, and I'm trying to figure that out. So hopefully you'll get your grades for exam 2 released soon. And then once I know what everyone's grades are and I do some calculations as a class as a whole, then you might be able to redo the second exam too, the same way you did the first. Any questions about exams? All right, really quickly, independent work on Monday. I had my dates crossed. So you don't have this week to finish independent work. You have this week and next week. So by the end of this week, you should have 90 points. By the end of next week, you should have 100. But I will say already, a lot of people did wait until the end to do it. So as you might imagine, I'm just getting flooded with papers. So it's going to take me a long time to grade your papers. And I just want you to know it's not because I'm slacking off or I don't care. I'm working hard, but I have to go through the ones that got there first. And again, I'm getting flooded. And like I mentioned before, I do run them through an AI detector and a plagiarism detector. And I don't trust them, but whenever I get a red flag on the red AI detector or the plagiarism detector, then I just really, really nitpick. I just have to be extra careful. So it's not like it says it's a red flag and I send it back to you and say, this says you faked it and you get a 0, right? I don't do that. But I go through very, very carefully, make sure everything you write is correct, make sure your sources are correct and all that. So again, I just apologize for that. Any questions about any of this or anything in general? All right, let's get moving. So again, the words today are not extra credit. They are credit. And the second word, official word, will be old. Anyway, so this is where we left off. And I kind of went through this slide pretty quickly. So I wanted to go back again. So we're talking about DNA replication, which you already know from previous semester or previous chapters. The DNA replication happens during S phase of interphase, right? That part's not new. The other thing you may not have known, and you definitely need to know, is what happens is you take the double helix, the original double helix, it gets opened up so that the code is exposed, right? And then there's an enzyme that we're going to talk about later that comes in and reads the code and brings in new nucleotides, right? So you can see this blue, all the blue ribbons, the old stuff. The gold ribbon is the new stuff. So what it's not showing is there's an enzyme here reading this, right? So it sees that there's an A here, so it's going to bring in a T. And covalently bond it to that. And then it's going to see there's a C here, so it's going to bring in a G and covalently bond it to that. That's how that works. There's a question on the study guide that'll most likely be on the exam. I'm trying to remember how it's worded. Basically, it's something like, what are you left with when you go through DNA replication? And the answer is you're left with 2 new double helix or 2 new double helices. And for each one strand is a new strand, and 1 strand is an old strand. You guys see that, right? Does that make sense? That will be a test question. You need to know that. All right. So are there any questions so far? We're still talking about this a little bit more, but that's what we have so far. All right. If you download the PowerPoint, you can watch this little animation that shows how this works. As I'm about to tell you a little bit later, the enzymes involved in DNA replication are also involved in fixing DNA, and that animation, if I remember correctly, shows both. This one is also an animation. It's a lot more simplified, and in my opinion, it makes more sense. So the other one I just showed you, it's more realistic, but this one's easier to follow. And then finally, this one too. This is another great animation. Also, I'll bring this up. This is a good reminder. If you're reading about this or writing about it for independent work, and you see a word that I haven't mentioned, just ignore it. So there's basically what I'm trying to tell you is we are doing like the Cliff Notes version of DNA replication. There's a lot of things your textbook and I am not talking about. If I remember correctly, definitely the old textbook, and I think your textbook too. I don't think they talk about leading strand, template strand. I don't think they talk about primase and helicase and a bunch of other enzymes. So again, if I don't mention it in here, you don't need to know it. So take notes. As a matter of fact, make sure you read your textbook. And again, If you see anything in the textbook and you're like, wait a minute, we had that discussion, I don't remember him saying this, feel free to meet me online for office hours, or just send me an email. Say, hey, the textbook mentioned this, you didn't. Do I need to know it? Anyway, all right. So what is the enzyme that does it? That will be a test question. The answer is DNA polymerase or DNA polymerases, because in all actuality, these are a team of enzymes working together, and collectively we just call them DNA polymerase or DNA polymerases. So that's what you need to know for the exam. DNA polymerase, that's the enzyme that does DNA replication. It is the enzyme that makes those bonds between the nucleotides of the new DNA strand. So again, to remind you of the stuff we talked about on Monday, DNA and RNA, they're made up of nucleotides that are covalently bond to each other, sugar phosphate backbone, right? So that's what does it. DNA polymerase does that. As I already showed you with that previous picture of the DNA double helix that had been split, and you had 2 more new ones coming, the incoming base pairs with a complement on the template. So again, like I said, and I'll show you again on the next picture, if there's an A, this enzyme says, okay, I better bring in a T, right? And if there's a T, it says, all right, I better bring in an A. If there's a C, it brings in a G, and if there's a G, it brings in a C, so on and so forth. It is fast and accurate. You do not need to know that, though, so I'll go ahead and put it next to that. I'm going to teach you about it, but that won't be a regular question. It might be on the study guide. You might get an extra credit question, but you do not need to know that. But yes, it is fast and accurate. How fast and accurate? It does about 50 nucleotides per second, and it makes about 1 mistake in every 1 billion bases, right? So it's very accurate, very fast. And like I mentioned earlier, it also helps repair DNA damage by toxic chemicals or anything, really. Any of these things listed, whatever, anything. If DNA is damaged, sometimes it can go in and make corrections. So any questions about that side? All right. And again, there's that picture I showed you. There it is again. Like I said, it's the code here that dictates which is coming in next, right? So this is a new strand being built, and the DNA polymerase knows that if that's an A, it needs to bring in a T. It knows if that's a C, it needs to bring in a G. And if that's a G, it needs to bring in a C, so on and so forth. All right. Again, as you already know, DNA replication happens during S phase of interphase. Where does it start? Actually, I wish I should have asked you before I showed you this slide, because I'd be curious to see where you thought it would start. To me, before I had taken any kind of biology class, I would assume something like this must start at the tip of the chromosome and work its way down. That's logical to me. But that's not actually how it works. DNA replication actually begins at sites called the origins of replication. You need to know that for the exam, because this exam is going to have a lot of questions about processes. Like, where does this start? Where does this end? So a lot of those questions are going to be on there. Where does it start? In this case, for DNA replication, it's origins of replication is the name of it. And there's more than one of them. So if you're looking at a whole chromosome, there's a bunch of them. So they get on there, they attach to the chromosomes, and then they all work from the different various origins of replication, which is quicker when you think about it. If you were copying a book, wouldn't it be quicker to like have a bunch of get a team together and say, all right, I'll copy chapter 1, you copy chapter 2, you copy chapter 3, so on and so forth, as opposed to one person starting at the beginning and copying towards the end, right? What happens is this thing, right? Here's some sites of replication, origins of replication. What's happening is the duplication is going both ways, right? Until it creates a bubble, until you have 2 new strands of DNA. But again, for the exam, the most important thing on this slide is that origins of replication. Well, and the S phase of interphase, but that's not new information. So the most important new information on here is knowing where this process starts. And it's the origins of replication. Again, the words today are not extra credit. The next one I will point an arrow to. So if you got your eyes closed, open your eyes, you need to write that word down. Your attendance will be based on sending in the words today. It's not extra credit today. Yeah. Anyway, if you download PowerPoint, you can see everything I just showed you, but it's animated. I highly recommend it. Or if I can find some other videos for you. Seeing this kind of stuff animated or in a video is much better because it really makes more sense to see it in action. Same thing. I don't know why it's like that. Oh yeah, and again, these are all videos, little clips you can watch. Once again, I'm not going to teach you about lagging strand and leading strand and 5' versus 3'. Those are all things you don't need to worry about. So if you're reading it in any other source, or even if your textbook, if you see some words or some concepts that I haven't taught you, you can just mental note, don't worry about it. You're not going to get tested on it. And if you do, it would be extra credit. All right, that was the easy part. We are now done with the first main bullet point, DNA structure and replication. Most of that was a review, right? You already knew about the structure of it because we learned that in the biological molecules portion earlier in the semester. You knew a little bit about the replication. You knew it happened in S phase of interphase. We learned that when we talked about mitosis and meiosis in the cell cycle. But here, let's get on to some new information. And in my opinion, this one's really, really important. So if you're going to understand a lot of drugs, if you're going to understand how viruses work, and how vaccines work, this is the information you need. So once again, I used this example on Monday. Somebody tried to say something to me online about how the mRNA vaccine is gene therapy and it messes up your DNA. I was trying to get to them, explain to me how that works, because it doesn't work that way. It goes from DNA to RNA to protein, not from RNA to DNA. But there are exceptions to that rule, and I figured, all right, I'll give this guy the benefit of the doubt. Tell me how that happens then. How do we go from RNA to DNA? And they couldn't explain it. Anyway, so this is important information. When we're done with this, you're not going to know that much. You're not going to know anything specifically about COVID-19 or its vaccine, but you'll know enough to where if you were to go and do your own research, I hate to say it like that, if you were to go read about some research people have done, you won't be starting from scratch, right? You'll understand the basics. So here we go. The DNA provides instructions to a cell, obviously, right? The cell itself is almost like a little factory, right? It keeps itself alive, and it has the instructions on how to do that. Those instructions are written in the DNA. But also, if you have a multicellular organism, such as ourselves, the DNA also provides an organism's whole genotype, right? And you need to know what a genotype is. I think I've already mentioned it before, but that is heritable information. It is the sequence of nucleotide bases in the DNA. So basically, it's all the A's and C's and T's and G's in your DNA. That's your genotype. It's all your genes. It's all the so-called junk protein that I haven't taught you about yet that you'll learn about later. So what comes from the genotype? The phenotype, right? So again, DNA provides all this. Obviously, the genes or the DNA provides the genotype, which also then results in the phenotype. Anyway, how does the DNA provide these instructions? This is what we're going to learn about. How an organism's genotype determines its phenotype. And again, you already know that the basics of it, right? We've already talked about complete dominance, incomplete dominance, codominance, all those things, right? Now we're going to learn exactly how it works. And again, here's a word you should already know. But once again, let's put it on the screen, see it in writing. A phenotype are the physical traits from the actions of a wide variety of proteins. So you can see the definition is a little bit different in the context of this chapter. All depends on the conversation you're having. So in the context of this chapter, again, it's the physical traits from the action of a wide variety of proteins. And I like that they included that from the wide variety of proteins parts, because we haven't mentioned that yet. But that is important, because think about it. I said earlier, or a lot I've been saying, DNA provides instructions on how to build proteins. But we're not all proteins, right? We're other stuff other than proteins. So then the question is, well, how then can DNA provide instructions for other stuff, like the size of our nose and, whether you're more likely to have breast cancer or not, right? This is not all proteins. So how does that work? Well, remember, proteins are enzymes of proteins. And basically, that's mostly what you're making, right? Your genes are making enzymes, and then those enzymes make other stuff. So for example, not what's written there, but I'll give you an example of the flower colors we were talking about for the pea flowers, right? The purple or white. Those weren't proteins, right? It wasn't a protein directly, like that's making that pigment, but there were enzymes, right? And those enzymes in those cells help make that purple color, right? So it's not like the gene actually coded for the color purple, right? The gene coded for a protein that then made the color purple. Anyway, that's not going to be important for the exam. I just wanted you to know how that works for your own knowledge. So some examples from the book. We have structural proteins that help make up the body of an organism. So yes, we do have some proteins. So obviously the genes do code for those proteins. But like I was getting at, more often than not, the genes code for enzymes. And it's the enzymes that make all these things, right? It's the enzymes that catalyze these chemical reactions that are necessary for life. So any questions about this slide? Making pretty good time. All right, here's some really important stuff. You need to know these two words: transcription and translation. As usual, I recommend studying the big picture and then adding in the details. So the first thing I think you should memorize is the fact that we go from transcription, or the first process is transcription, the second process is translation. Memorize that, and then start adding in the details. Another thing you can memorize that's related, and it was one of the first slides I showed you in this conversation, is that the process goes from DNA to RNA to protein. Memorize that, right? DNA to RNA is transcription, RNA to protein is translation. Memorize that. Have that understood. Then, once you understand the basics, then start adding the details. In a perfect world, this would be the end of the lecture. Right? We would have a really short lecture, and I'd send you home, and then we would meet tomorrow instead of Friday. And then once you had that memorized, then I'd add a little bit more information, and we'd have a short lecture. And then I'd send you home, and we'd meet the next day. But it's not a perfect world. Anyway, the relationship between genes and proteins are one of information flow, which is one of those things I talked about way back in chapter 1. I said that's something we're going to see a lot of in this semester, and it's back, information flow. That's not going to be a question or anything. So again, as you should know, the function of a DNA gene is to dictate the production of a polypeptide. What's another way of saying that is a gene provides instructions on how to build a protein. You need to know that. So again, DNA to RNA to protein. DNA to RNA is transcription. RNA to protein is translation. Any questions so far? All right. So first thing we're going to do is an overview. And again, I'll remind you, if you're following along with the online textbook, I'm doing it in a slightly different order. I'm giving you the same information. I'm just changing the order because I like this order better. It's from your old textbook. And what we're going to do is an overview before we start doing the details, which is, again, how I recommend studying. So the information in the DNA is transcribed to RNA. Like I said, you already need to know that. That's the first thing you need to know. Going from DNA to RNA is called transcription. And then that RNA is then translated into proteins. And if we're going to get real specific, technically it's translated into a polypeptide, which then folds into proteins. And I don't think I'm going to get that specific in this exam. You needed to know that for the first exam, because when we talked about biological molecules, we talked about the relationship between a polypeptide and a protein, right? A polypeptide, just a long string of amino acids, right? And it's not considered a protein until that thing is folded into its correct shape. Just like you might have a long string of yarn, it's just considered yarn until you've, crocheted into a scarf. At that point, it's still yarn, but it is now also considered a scarf because it is in its correct shape. Same thing here. Anyway, any questions so far? Here's what I'm saying. Again, overview. Inside your nucleus, you have some DNA. That DNA is transcribed into RNA. And this also happens inside the nucleus, which we'll talk about later. For now, don't worry about that. That RNA then goes through some changes. It leaves the nucleus and then is translated into a protein. So there's a few test questions there, and you don't necessarily need to write them down yet because I'm going to talk about them later. But you could say, again, what's the process? What are the steps? And you need to know DNA to RNA to protein. And then you need to know a good test question is like, what do you call going from DNA to RNA? Transcription. What do you call going from RNA to protein? Translation. Another test question is, where does transcription happen? Happens in the nucleus. Where does translation happen? Happens outside the nucleus, right? There's a few test questions there. Once again, if you download the PowerPoint, you can watch this little animation. So I keep saying transcribe and translate. And these are words that you would also use in just regular everyday life, right? Almost as if you're talking about a language. So if you had a boss and they had an employee and the boss was like, well, you probably wouldn't need to do this with technology anymore, but the boss might say, all right, I'm going to write a speech. I'm going to say the speech. I need you to transcribe it, right? That just means you need to write it down, right? But then maybe they go to the UN and he said the speech in Spanish, but it needs to be translated to German, right? So you need to change it from one language to the other. That's translation, right? Same kind of situation. So in a sense, you could almost think about this whole thing as a language. So let's try to approach it from that point of view. Let's think of this as a language. And it is in a sense, right? Because I keep saying the DNA provides instructions on how to build proteins. Well, that's a language, right? The DNA is saying in some sort of language, this is how you build a protein. So what is the language of nucleic acids? Well, the first thing you already know is this bullet point. We already talked about this on Monday. For DNA, the language is the sequence of nucleotide bases. So the same way our English language goes from A to Z, right? I mean, every letter in between. The DNA language is just A, T, C, and G. That's the language, right? The English language has all the letters. This language has those 4 letters. And a musical, like what do you call it? Musical notes? What's that? 12 keys? A, B, C, D, E, F, G, A, B, C. Anyway, yeah. So 4 letters, right, for this language. A typical gene has thousands of nucleotides. Let me put an X through that. That's just for your own knowledge. I'm not going to ask you the size of a typical gene. That is a good independent work question, especially if you're not good at writing long papers or if you're not good at summarizing, so you're worried that you're going to get in trouble for plagiarizing. Just write a paper that says this. I was wondering how many nucleotides a typical gene is, and according to this source, a typical gene is this many nucleotides, right? I'm saying thousands, but of course, depending on your source, you might get a slightly different answer, maybe a little bit more precise. Not that you need to know it, but it's a good independent work question. Anyway, that DNA, the language, right? This words written in this language of the DNA are then transcribed to RNA, right? So think about it again. I'm saying this is transcribed. It's written down. So we haven't necessarily changed the language. We're still in this language of nucleotides. The A's, the T's, the C's, and the G's. Of course, for RNA, to remind you, RNAs don't have T's. They have U's instead, but it's still the same language, right? Anyway, any questions so far? All right. That was transcription. And remember, this is an overview. We're going to come back and talk about all this in more detail. Translation is the conversion, right? Just like anything else. When you translate, you're changing from one language to the other. In this case, we're changing this from the nucleic acid language, there's A's and C's and T's and G's, to the polypeptide language. So to remind you from what you learned way back in chapter 2, God bless you, there are 20 amino acids, right? So the DNA language, the nucleotide language consists of 4 letters, A's, C's, T's, and G's. The polypeptide language, if you will, consists of 20 letters, if you want to call them that. 20 different amino acids, right? So it's the sequence, it's those letters written in the RNA that says which amino acid needs to be attached. Because remember, a protein or a polypeptide is just a bunch of amino acids covalently bonded to each other. It's just a long string of amino acids. That's all it is. And it's the RNA that says, all right, first bring me this amino acid. Okay, now bring me the next amino acid. And it gets attached, right? And then it says, okay, bring me the other amino acid. That's all it is. It's really that simple. Of course, we're going to make it a little bit more complicated, but at this level, it's pretty simple, right? Let's see. Next word for attendance. Again, I'll point to it. I just crossed through it on accident. I was trying to point to it. I'll just say it. Acid is the next word for attendance. Acid. As in something with a low pH or with a lot of hydrogen plus ions. Anyway, any questions about that side? All right, here's kind of a picture of everything we've said. There's a strand of DNA. Obviously, it's going to be a lot longer than that. If this was to scale, it would be huge. We've zoomed into a certain portion of it. We can see 3 genes. And we're going to zoom in even closer to that gene 3, whatever it may be. So this is just one small chunk of that whole gene. And you can see here's the DNA strand, right? Here's the words, here's the letters. And these letters get transcribed into RNA, as you already know, is base pairing rules, right? So A's always go with T. There is no T in the RNA, there's only U. So A's go with U, C's go with G, G's go with C. Yeah. Anyway. And then that's your RNA, right? And then those RNA letters transcribe or translate into amino acids, right? So whatever UUU is, that's like saying, bring me a square amino acid. GGC says, bring me a circle amino acid. GGU says, bring me a triangle amino acid. Of course, this is just an example. There are no like square amino acids, circle amino acids. It's just easier to visualize. Does this all make sense? So again, you have all these letters here. It gets transcribed, so they're just written down there. We haven't changed languages yet. Then that gets translated into that. Does that make sense? All right, so I'm going to go back to an analogy I've used before. You can think of your chromosomes as books, right? You have 23 sets of books in every one of your cells, right? 23 sets of books, 23 pairs of books. And each one of those, remember that's supposed to be like a chromosome, each one of those books has a bunch of instructions on how to build a protein. But you only have, your cell only has that one master copy. So imagine if this was WVSU's campus, and we had like a library where all the workers go, and they have instructions on how to build stuff on campus that we need, right? Like we need a bench, we need some seats installed, whatever it is that anybody on WVSU's campus might need. Imagine there's only the one copy, or there's only that one original set of books on the campus. Does that make sense? The one set. Also imagine there's no internet. That helps too, because otherwise you could just look this stuff up. So anyway, you've got your set of instructions, and they keep it at this library or whatever. So now whenever a worker needs to build a bench or whatever, they go to the library, they find the chapter they need, which would be the gene, and they see what's in there and they write it down, right? Because they have the main copy. They don't want to take the main only copy out of the library and take it and work, go out and build a bench reading those instructions, because they might destroy their only copy. Same thing here, right? We don't want the DNA to leave the nucleus. It's safe in the nucleus. So instead of taking the whole chromosome and taking it out of the nucleus, we've focused on the one gene that we need, write it down, and then it can leave the nucleus and get translated. Or in that analogy, then the construction worker can take those instructions that he wrote down, take them out to wherever he needs to go, and translate that into a bench, right? So he'll read it and say, all right, step 1, get a 2x4. Okay, step 2, attach that 2x4 to this 2x4 using this, right? He's translating the words that are written in English into a bench. Anyway, any questions about this so far? Again, we're just scratching the surface. We're going to go into more detail later. The flow of information from gene to protein is based on a triplet code. Another way of saying that is, again, remember, the nucleic acid language comprises of 4 letters, unlike the English language, for example, right? We're dealing with 4 letters. And another way that this language is different is all the words in this language are 3-letter words, right? That's what I mean by triplet code. So unlike the English language, right? So you could have a word like A, which has one letter, or you could have a word like supercalifragilisticexpialidocious. I don't even know how many letters are in that word. But the point is, there's a lot of variation in the English language, right? In this language, there's not a lot of variation. There's only the 4 letters, right? A, T, C, or G. And for all the words in this language, all the words are only 3 letters long, right? So there are no 1-letter words or 2-letter words or 4-letter words or 5 or whatever. They're all 3 letters long. So the instructions for the amino acid sequence that are written in the DNA and then the RNA serve as a series of words called codons. They are a series of words called codons. So in other words, the triplet code, another word for that is codons. I'm not going to test you and ask you what a codon is, but I'm going to use that word a lot. Now that you know what it is, that's the word I'm going to use from now on. A codon. A codon in DNA, so the word in the DNA is transcribed into the complementary codon in the RNA. And again, another way of saying that is the 3-letter word that's in the DNA is just written down as RNA, right? So we still have that 3-letter word. Then that 3-letter word, that RNA codon, is translated into amino acids that form polypeptides. So again, those 3-letter words, each 3-letter word just says, bring me this amino acid, bring me that amino acid, right? Because there's 20 different amino acids that they're working from. And each one of those 3-letter words says, bring me this particular amino acid. Big picture here, the most important thing you need to know for this slide is the word codon, because we're going to cover all of this later. And I'm going to use the word codon when we cover it later. Any questions about that? Doing pretty good on time. We already looked at that. Let me skip forward. So before we can get into the process of transcription and translation and all that, there's some other background information you need to know. So let's talk about the genetic code a little bit more. We've already started talking about it. Let's talk about it a little bit more. The genetic code is the set of rules that converts RNA sequence to amino acid sequence. So that whole thing where I said that the codons for RNA code for particular amino acids, I'm saying it again. And now I'm telling you there's something called a genetic code. It's just a set of rules. You don't need to know this, but your book included it, so so did I. But first of all, again, let's take a step back. This language consists of 4 letters, A's, C's, T's, and G's, and put together in different ways, just like any other language with an alphabet. This language has words, and these words, they're always 3 letters long. And now I'm giving you one more set of information, or one more bit of information. This language only has 64 words in it. So it's a lot different than the English language, right? There's like millions of words in the English language. But for this, there's only 64. Let me put a question mark next to this. If I was going to ask you anything where that X is, that might be the one thing I would ask you. How many words are there in this language? There's 64 words. 64 triplets, 64 codons, however you want to say it. But then for your own knowledge, and I'm certainly not going to ask you this, 61 of those words actually say, bring me this amino acid, bring me that amino acid, whatever it is. And 3 of those words say, all right, we're done, stop the process. So again, most likely I won't ask you anything that's been crossed out there, but if I do, it would be that first main bullet point. And you would probably be more likely to see that in the good grief, I can't think right now. In the exam. So a given RNA triplet always specifies a given amino acid. I think this is pretty fascinating too, and this is what we're about to get into. Doesn't matter what the organism is. It could be bacteria, could be a dinosaur, if you could somehow get dinosaur DNA, could be a human, could be a yeast, could be a cat, whatever it is. If it's alive, there's been no exceptions to this rule that we've ever found. The code is always the same. So in your genes, if you have a word that says, bring me this particular amino acid, that gene for any other human and any other living thing on Earth would say, bring me that particular amino acid. And here's how it works. And you might need to know this for the exam. There might be 1 or 2 questions on it like this. And it might look complicated, but it's not. So I might give you a word and say, okay, or a codon and say, what amino acid is this coding for? I'm not even going to look at the wheel yet. I'm just going to make up a codon. Let's say I give you, it'll say on the exam, what is CCG, right? Translate CCG. So, all right, we would start here. Wow. I don't want to get in front of the screen because the camera's messed up. Let's do it this way. We'll start here in the middle, right? Like, okay, there's a C. All right, so now I go next and find a C here. Okay, there's another C. Now I need to find the G, and it is right here. So the codon CCG translates into the amino acid proline. And that's how that would work on the exam. I would give you this wheel, and I would ask you to translate something. Does that make sense? It should be pretty simple. You just start in the middle and work your way out. And I'll give you one more example. Let's say, I don't know, UCA. Why not? Let's see what that translates to. So UCA. Good job, right? So again, start at the U, C, A, serine. And that's it. All you have to do is memorize this. No, I'm just kidding. You don't have to memorize this. If you have any questions, again, I would give you this, and then I would give you the codon or codons. It could be more than one, and say, please translate that. So anyway, any questions about that? All right, pretty simple stuff. And like I already alluded to earlier, the genetic code is universal. And this is what I love about it. It's so fascinating to me. It's shared by the simplest bacteria to the most complex plants and animals, right? Everything on Earth shares the same code. So for that reason, one species can produce protein from another by transplanting the DNA. And that's, if you're into that, there would be so much stuff to look into for independent work for that. For example, they've had goats where they put spider genes into the goat's genome. So when the goat produces milk, that milk also produces spider silk. All because they changed the gene, they put the gene from one place to the other. There's a rice called super rice, I think it's called. Super rice? It might be called super rice. Golden rice. Anyway, it's regular old rice, but what they did, since a lot of people in starving parts of the world, that's their staple is rice, but they're still not getting a lot of nutrition. They took the gene to make vitamin A out of carrots. They put it into the rice, and now this rice produces vitamin A. Right? There's all kinds of crazy stuff going on. I'll give you some more examples with some pictures showing up. I'd like to do this. Like my brother lives in Seattle, and marijuana is legal. I think it'd be cool to get the genes responsible for making THC and putting them into edible foods. So like your salsa could get you high. If you're into that, I'm not saying I'm into it. I'm just saying it's a good money-making venture, right? A salsa that gets you high by using the actual tomato or pizza sauce or whatever. Anyway, there's a lot of stuff like that. That's what GMOs are, basically. And you can look that up for independent work. Everyone's scared of GMOs. I can see how some of them are scary, like BT corn produces its own pesticide. Or is it pesticide? Yeah, pesticide. It's completely safe to humans, but I can see how that's scary because it sounds scary. But this other stuff, like the golden rice, that's not scary. There's an apple that they produce that doesn't bruise or turn brown because, well, this is kind of the opposite. They took the gene out. But anyway, here's some examples. You might tell what's up with this pig in the middle as compared to the other two. This is a real picture. Can you tell what's wrong with it or how it's different? It's a little bit hard to tell. It is yellow because it is glowing. That is a pig that glows in the dark. They took genes from a jellyfish that allow it to glow, and they put it into that genome, and now that pig glows in the dark. And don't get me wrong, this isn't a bunch of mad scientist stuff. People are like, I want to see if we can make some glowing pigs. They do that stuff for a reason. You can look that up for independent work too. But basically, if you want to see how a gene is expressed, you can attach a jellyfish gene to that gene. So then you can see everywhere that gene is expressed because literally it'll glow. Not that you need to know this, but for example, not all genes are expressed in your body equally. Some parts of your body express certain genes more than others. And then again, in that case, you could do that. Anyway, this same case here. Here we have a glowing, I think it's a mouse. It's a baby, whatever it is, baby mouse, baby rat. It's glowing. And then this picture down here is fake at the bottom left. But I have read about people wanting to do it. They want to take, again, the same genes from jellyfish that make them glow and put them into trees so that the trees glow. And then you could eliminate, in theory, you could eliminate street lights. I have issues with that, but that's a whole different discussion. Anyway, any questions about the genetic code? All right. I feel like we can start and finish transcription. That way on Friday we can just jump into translation. From is the next word for attendance. From. So again, transcription is the transfer of genetic information from DNA to RNA. So if you're making flashcards, you could literally just put transcription on one side of the flashcard. On the other, put DNA, arrow, RNA, right? Transcription is going from DNA to RNA. That's what you need to know. As you already know, well, no, I guess technically you don't know this. I haven't taught you this. So similarly to the way we do DNA replication, similar here, the DNA strand opens up because that's where the code is, right? That double helix, those bases are hydrogen bonded to themselves. But we need to get to those bases because that's the code. So we have to open up the double helix to expose the code. Another thing, I'm going to put it next to this because you don't need to know this, but I just wanted you to know because your book talks about it. When you open that DNA strand, and it's going to be a horrible picture of it. There's your DNA strand, and then it opened up right there. Good grief, that's horrible. Right, there's the little bases hanging off the DNA strand. That's so horrible. Anyway, when we do that, only one of these, let's say maybe that one. That one's the one that's serving as the template, right? That's the one that's the code. The other one is just ignored. That's just for your own knowledge. We don't need to go that deep when it comes to the exam. The RNA nucleotides take their place along the DNA template by forming hydrogen bonds with the DNA nucleotide bases. I'm not even going to go into that much detail either, so you don't need to know that either. Again, what you need to know, transcription is going from DNA to RNA. So we're taking the code in the DNA and we're writing it down as a strand of RNA. That's what we're doing, right? We're transcribing it. To do that, again, we have to open it up because we have to expose the code. Any questions so far? We're going to talk more about this. And the reason I have a pen is because, again, I want to reinforce this idea of transcription. We're just writing something down. We're copying it from one to the other. We're not translating. We're not changing languages. The RNA nucleotides follow the same base pairing rules. So again, we're doing A's to C, A's and T's, C's and G's, right? Except again, RNA does not have a T, it has a U. So for example, if there was a DNA strand on the exam that said transcribe this, and it said, I don't know, CCT, CCT, I don't know, AGG. You might get a question like this on the exam, and it says, all right, transcribe that. All right, so C goes to a G, C goes to a G, T goes to an A goes to a T, except again, we're doing RNA. T, there is no T, there's a U instead. G goes with a C, and G goes with a C. Sorry, it's all squiggly, but I can't, it's out of my control. So that's the kind of question you would get on the exam. You guys comfortable with that? All you have to remember is that A's go with T, C's goes with G, and that there is no T in RNA, there's only a U. If you can remember that, there's nothing hard about this. Another thing you need to know for this exam is what is the process, not what it's called, what enzyme does this, and it is RNA polymerase. So there you go. There's two polymerases I've taught you about today. And it's in the name, but you need to know them both. You'll get questions on both, and hopefully no one screws it up because it's in the name. DNA polymerase makes new DNA in the process of DNA replication. RNA polymerase makes RNA in the process of transcription. And this picture is actually what it looks like. So right here at the bottom, this long thing, that's a strand of DNA, as you can probably tell. This big old blob that's attached to it, that's the RNA polymerase. This little squiggly tail coming out of it, that's the RNA that it's making. And notice too, because I don't remember if your textbook talks about this or not, that RNA polymerase is actually different colors because, and again, this is for your own knowledge, I'm not going to test you on this, that RNA polymerase is actually a team of proteins. It's not just one protein, it's a team of proteins. But for this 100 level non-biology major class, we just call it RNA polymerase and we leave it at that. So any questions about that side? All right, see if we can finish this up. This is what it looks like. Again, zooming in. The RNA polymerase is attached. The DNA has opened up to read the code, right? We're filling this in. We're making this new strand of RNA, one nucleotide at a time. We're reading the code, right? The T codes for an A. A usually codes for a T, but there is no T, so it brings in a U. Again, A brings in another U, and that's it. It just reads the code, brings in the next nucleotide, covalently bonds it, and then the DNA closes back up. Process is done. Any questions about that side? All right, here we go. Let's make it more complicated, right? That was too easy. It's biology. We have to make the test hard. I'm just kidding. That wasn't the reason for this. But transcription itself is broken down into 3 stages, and you need to know these 3 stages in their order and what they do. Of course, the names kind of give it away, but the stages are initiation, elongation, and termination. Initiation of transcription, RNA elongation, and termination of transcription. I put those 3 words in bold, though, because, again, if you're just studying like with flashcards, that will suffice. 3 stages of transcription, initiation, elongation, termination. If you can memorize that, you'll be fine. And we're going to separate all 3 separately. But again, I recommend when you're studying these things, study the big picture and then starting at it in details. So before you start studying what I'm about to teach you, first memorize the fact that transcription is broken down into those 3 stages. Once you have that memorized, then start worrying about what they are. Of course, again, this is not a perfect world, so I can't just leave you with that and have you study it and then come back. I've got to move forward. So let's move forward. Initiation, that's step 1. Initiation starts at a place called the promoter, and you need to know that. There will be a test question that says, where does transcription start? It starts at the promoter. So the same way DNA replication starts at the origins of replication, transcription starts at the promoter. If you know that, then you're good. But let's talk about these other things that I'm not going to ask you about. The promoter is located at the beginning of a gene. That should make sense. If you need this whole gene, you don't want a promoter in the middle of the gene, right? Because then it's only going to read half the gene. So it should make sense that the promoter is at the beginning of the gene. Obviously, if the RNA polymerase is the thing that's doing the job, obviously that's where it's going to attach, is at the promoter. So what is initiation other than the first step? It's just simply the attachment of the RNA polymerase to the promoter. That's all that happens for the initiation, right? Initiation is the first step. It's when the RNA polymerase attaches to the promoter. And that is where the process starts. And that is where our lecture ends. So I will see you in class on Friday. Or I guess that's it. Yeah. I will not be in lab today, but the TA will be. Any questions? Well, let's do one last word for attendance. And that is Terminator. Since that's what's on the picture right now, is a Terminator. And that's it. Last word for attendance is Terminator. I'll see you guys on Friday. Or I'll be online for office hours if you need me, assuming I have my computer.